Okay, try, Taylor.
All right, hopefully you can see the screen.
Yes.
Great. All right, let's begin. Thank you everyone for attending this talk. My name is Taylor Shaw. I'm from the University of Freiburg in Germany, but today I'll be sharing with you a project I did with colleagues in Sweden, which was to acoustically survey a boreal forest during winter. So why monitor in winter? This is quite a dense slide. I won't make you read all of it, but essentially this is when it's an opportunity to study the resident birds, which are indicator species. And as Alessia said a few talks ago, that they're indicators of forest naturalness. And we have a lot of primary cavity nesters in this group. So these are very important birds and of high conservation interest. However, we know very little about what they do at these high latitudes during winter. This is also a really critical survival period for them because of food shortages, lower temperatures, and in the case of forests with deciduous vegetation, drastically altered habitat architecture. We also know that these residents are very sensitive to forestry-induced changes in their habitat. We've observed local extinctions and abundance declines in Scandinavia. So take all of this together, we wanted to investigate how a simplified forest structure may affect the winter survival potential of these birds of high conservation interest. So the objectives of this study were to identify links between bird diversity and their use of forest structures in winter. Although because we weren't sure what we'd get, because normally birds are surveyed during the breeding season, we wanted to use two different methods. We first used the traditional way, which is point count surveys. And secondly, we used acoustic monitoring techniques. So within the acoustic monitoring, we also employed sort of a hybrid approach. So we used bioacoustics to get individual species identifications, and then we also computed ecoacoustic indices. It should be noted here, and that we've seen this in a lot of other talks and in my previous research, that often point counts are used to ground truth acoustic indices. But in this case, we ended up having two different ways to ground truth the acoustic indices with point counts and bioacoustic ID. So to do this, it was a three-step approach. The first thing we wanted to do was compare point counts with bioacoustic identifications. And there's only a limited way you can do that because point counts reveal richness and abundance, but you can't reliably estimate abundance with bioacoustic methods. So the only metric we can compare is richness. And don't worry about what this means because we'll be revisiting this slide a couple times and I'll explain this variable later on. But essentially, step one is to compare the only metric we can, which is species richness. The second metric is to take, or the second step is to take all of the metrics that we get. And here we have metrics from point counts. Here are all of the metrics that we'll be able to glean from bioacoustic identification. And we're going to see how each of these metrics responds to the vegetation in our study site. So the structural heterogeneity, the deadwood, for example. And then the third step, after we've established the relationship between birds and the vegetation, is to see how well these acoustic indices reflect those relationships as potential proxies. Okay, so our study site was in Färna Eco Park. It's a conservation area that's dominated by spruce, Scots pine, birch, and aspen. Some of these stands are full monoculture spruce, but they go all the way up to 70% deciduous trees with birch and aspen. And the more deciduous coniferous mixed stands have higher structural complexity. So it's within that framework that we set out to identify 20 plots with a 50-meter radius around a GPS point, but we also ensured that the forest classification, so the same mixture of tree species and the same ratio extended beyond the 50 meters because, of course, recorders record for that further distance. We ended up with 19 sites altogether. You can see this is a very patchy mosaic area with water bodies, water courses, and along this axis and this axis, we have roads, and the orange is a little bit of settlements. So it's an area of high conservation value, but has a lot going on. I'm going to gloss over this just for the sake of time, but essentially we did 6 circular subplots to do the vegetation surveys where all of the living and standing and lying deadwood trees, their species identity, their DBH, their height, all of the metrics we could collect over 8 centimeters dBH. So everything that was, not very, very young, we recorded. And from that, we used actually from colleagues at our university in Freiburg, building off of their work, we computed all of these metrics that when you take them together, they describe stand structural diversity that's ecologically relevant, which has to do with the variation in the size, the height, the age of these woodlands. But because it's so many variables, we condensed it into a PCA. The first axis, axis 1 here, tends to represent structural heterogeneity. Mostly, it's a little small, I apologize, but this is the standard deviation of the dBH, the standard deviation of the height, and so on. The second axis fully captures the deadwood volume of both standing and lying deadwood. So that fell together nicely, and that's the structural variables that we were using. For the point count, my colleague Gregor Mikuzinski did all of the point count surveys himself. He visited each site twice and for 20 minutes documented all birds seen and heard with no distance limit. So the important thing to take away here is that he sampled 20 minutes per site. And of course, as I said before, the two variables that we get from this are species richness and abundance. For the acoustic survey, we sampled 1 minute every 10 minutes, all day, all night for the winter months, the same months that Gregor was doing the point count surveys. Winter is a very geophonic season, especially at these high latitudes. So first we cleaned the wind, the rain, and also we had some device glitches due to the changing temperatures, which we were able to clean out. After we did that, we had a clean data set from which we randomly sampled 2 sets of files. The reason we randomly sampled 2 different sets is because we had the resources to identify 200 1-minute files per site for bioacoustic identification. But we also did a subset of 40 minutes so that we could directly compare the sampling effort between point counts and bioacoustic identification. So what we get from all of that bioacoustic work is 45% of the files contain bird vocalizations. And from that, we were able to compute 2 species richness variables. Species or richness equal is the one equal to the point counts. Richness max is the one that basically was able to identify more minutes than you were able to do in the field if you're doing point counts. Now, you can't estimate abundance based on bioacoustic work, but you can get different information, which proved to be very useful. So the first metric here is number of visits, and that is the cumulative number of bird visits, all species pools per site. This second metric is number of flocks. So it's the number of flocking events we were able to observe per site. And number of multiple birds, it's kind of a clunky name, but it's the number of 1-minute files per site that contain different bird species within that 1 minute. And all of these are basically a metric of habitat quality. Because if we're continuously sampling every 10 minutes all the time, and these habitats are more attractive because of food availability, because of shelter, maybe hiding from predators, birds are going to spend more time there. And the more time they spend, the more chances that once every 10 minutes, we're going to capture something that they're doing. And so the higher that these metrics are, the more it's an indicator of habitat quality and time spent, basically habitat selection. For the ecoacoustic data, we computed 6 of the most popular acoustic indices. I'm sure many, if not all of you, are familiar with these. And we computed daytime means per plot. Okay, so back to this slide. The first thing we're going to do is just compare the species richness from point counts, which you see here, and the 2 richness values we get from bioacoustic identification. And what we see is, with a higher sampling effort, unsurprisingly, species richness for bioacoustic ID is way higher. We also see that if you compare 40 minutes per site with bioacoustic ID and 40 minutes per point count, it's higher, but not statistically significant. If we go a little deeper, I can give a few descriptive statistics. The point counts yielded 30 unique species. The recordings with 40 minutes recorded 25, and the recordings with 200 minutes per site recorded 37 unique species. Interestingly, the point count survey recorded 5 species that none of the recorders picked up, but the recorders picked up 12 species uniquely that the point counts did not. And then there were 25 species that both methods captured. When we get a little bit deeper into the composition of these groups, we see something quite interesting. So there are some birds in this both section here on the left, like the great spotted woodpecker, where they were equally observed by both methods. And by that, I have 2 numbers here, which I can describe. The first number is how many sites the point counts observed this species. So 12 means 12 of the 19 sites, the point counts observed a great spotted woodpecker. On 14 of the sites, the bioacoustic ID with the same sampling time recorded this bird. And even though the bioacoustic ID is slightly higher, I would consider these more or less equal. However, there were some species that were very, very present in all or most of the plots, but the point counts failed to capture them. So we have here the goldcrest, for example, was present on every single plot, but the point counts only detected them on 4 sites. For the tree creeper, they were present on 17, but only 1 site, the tree creeper, was observed by a human. So what we see here is that the bioacoustic ID can capture some birds that the point counts are not, but we didn't see the reverse. We didn't see that there was certain species that the point count was regularly picking up that the bioacoustic ID wasn't, which I'm sure will yield a heated debate at some point between ornithologists and ecoacousticians. The second piece is to test how all of these metrics together, so these are from the point count, and these are from bioacoustic identification, how they respond to the PCA axes that I described before about the vegetation. So what we have here is this first column is the PCA axis 1, structural heterogeneity, and this is deadwood volume. For the point count, the dotted line is non-significant. We see a very flat response. The point count, the richness values from the point count do not respond to forest structure. However, the richness of 40 minutes and the richness of 200 minutes does significantly respond, which I find this quite interesting because as you see here, the 40 minutes from the bioacoustic ID is statistically similar to the 40 minutes of point count, but here you see it's responding while the point count is not. So we're getting a little bit finer information here and the gradient is slightly larger. When we look at the other metrics, abundance is from point count, and then these are the new bioacoustic metrics. We see that 2 of the 3 bioacoustic metrics significantly responded to the structural heterogeneity of the forest. Number of flocks didn't, and abundance from the point counts didn't. So what we have here is none of the metrics of the point counts responded to any of our structural metrics. We have, however, almost all but 1, every single bioacoustic metric is responding to the structural heterogeneity. I'd like to point out that all of these slopes are pointing downward, but this is actually because our PCA loaded in the negative direction. So this actually shows that the higher structural heterogeneity, the higher number of visits, even though the graph is counterintuitive. Also, interestingly, nothing responded to deadwood. Okay. The third step to test how the ecoacoustic indices reflect these patterns. We tested each of the indices here in response to each of these metrics via GLMs, and this is what we get. Highly significant was the acoustic complexity index with all of the same variables that responded to structure. So this is from point count here. And here, abundance is also from the point count. We're not seeing these metrics responding to the structure, and we're not seeing the acoustic indices responding to these metrics. However, we are seeing this pattern follow all the way through from the structure to the bird community and the bird activity, all the way to the ACI. We see the same pattern exactly with the bioacoustic index. And I find this really interesting. I know that this is only winter, which is sort of untapped territory. However, if we had simply assumed that a point count was a sufficient way to monitor, we would have taken this data and we would have tested a correlation or a linear model from the acoustic index in response to this richness, and we would have assumed there was no relationship. We also would have assumed there was no significant metrics that we could record about the stand structural heterogeneity important for birds in winter. But all of this information is here, and there are clear patterns that we're finding that if we had only used point counts, we would not have seen them. So yes, we have acoustic indices. We've been able to sort of track it back to the bird assemblage and activity and back to the forest structure. Some takeaways from this are that point counts are not always an adequate ground truth for acoustic indices. For example, this study in a boreal forest in winter. Acoustic indices, we've also seen, are not a direct proxy for one metric, for example, bird richness, but they represent multiple aspects of the bird assemblage and multiple aspects of their activity. For example, all of those new metrics that I presented today, such as the number of visits and the number of multiple birds per file. The ACI and the bioacoustic index were strong proxies for richness and activity for this site, and they also reflect responses of these birds to the forest structure, essentially habitat selection. And then the other 4 indices did not respond, and we consider this to be a matter of robustness because only 45% of the files contained bird song, bird calls. The other 55% of the files were silent or windy. And we just think that for certain indices, they need to have a higher threshold of calls within the full batch in order for these indices to really be responding at the average level, because we were condensing these metrics into plot averages. So in conclusion, the bioacoustic approach is quite useful and reveals different and additional metrics for bird diversity and activity. What I thought was quite interesting was, despite the large home ranges of birds in winter, we can still observe stand level structure selection. And the ecoacoustic approach is also useful, for example, in this case, the ACI and the bioacoustic index. But these indices are site specific, and their robustness needs to be tested at these sites over different seasons and multiple years before I think you could really rely on them as indicators alone without this other information. So with that, I would like to thank everybody who participated and supported this work from 2 universities, and I would be glad to take any questions.
So thank you. We can open discussion. I see one hand raised, Bernie Kraus. Please, Bernie.
Yes. Hello, Taylor, and thank you for that. I came a little bit late into your discussion, but I have one question. Did you test different recording models to see if different systems picked up a wider range of vocalizations?
No, that's an interesting question. For this study, we used the same device throughout all of the sites just to standardize it.
And that was?
That was a Lunitronic Soundscape Explorer prototype that we had from before.
Okay, go ahead.
I was going to say, we've done other research where we've tested the SM4 and the Lunitronic device, and they've definitely been able to capture different frequencies at wider ranges, but not for this study.
And a different range of range of vocalizations as well. What I mean by that is they were able to pick up. Some systems are able to pick up a much wider range of sound from a much further distance, and you might find that testing different systems just to see which ones work, because they'll give you a different result. Mm-hmm. And so the result of one system may not be as conclusive as you want to have in the final data.
That's true, but when you standardize it, you can say that, okay, if you assume that the microphones are of the similar quality and all of these things, and you're orienting them all in the same direction out of the prevailing wind direction, for example, then you can say that given that recorder detection space is in constant fluctuation, it's as standardized as it can be. So within the same space around every site center, this is what you're getting. And if you use different devices, you might be expanding that detection space range or shortening it. And therefore you're capturing more vegetation in or outside of the plot. So at least for this study, I consider it an advantage that we only used one device.
Okay.
Especially because our plots, there were 50-meter radii,
Right.
and then 100 meters around, it was the same forest classification, but because this landscape is very mosaic, past that, you could enter a very different forest type. And so playing with the detection space range was already something of a concern for us, which is why we were so excited to develop a couple new metrics, because we thought we could just get almost every bird in every site, at least if they're flying around and trying out different types of stands. It's about how long they were spending in each of these that we thought would be more revealing or interesting.
Yeah. Okay, thank you.
Thank you.
Okay, we go through the